Aloha everyone, my name is Christine Flauta. I work for the state of Hawaii in the Division of Forestry and Wildlife under the Native Ecosystem Protection and Management Program. Today I'll be talking about a comprehensive method for detecting and controlling Angiopteris evecta in inaccessible terrain on Oahu, Hawaii. So just a little overview today, I'll be talking about why is Angiv a problem in Hawaii, the methods of control and problems that we've encountered with each control, our results, and our next steps for the future of this project. So why is Angiv a problem? So as you all may know, it is not native to Hawaii. It is actually found more in Australia, New Zealand, and kind of the South Polynesian Islands. And sadly, not a lot is known about Angiv. So we don't know the longevity of the spores, how long they can last in the soil. We don't know how long these plants actually do live. And there just hasn't been a lot of research done to know what kind of control can be done for Angiv. And just like any other invasive species here in Hawaii, it does displace and outcompete many of our native species, which then decreases the biodiversity and the integrity of our Hawaiian native forest. And with this decrease of biodiversity, it does hinder suitable habitat for some of our native plants and animals like our kahuli tree snail, our opaiula, which is our native shrimp, or some of our native endemic plants. So here we have a map of Oahu, and it's just to give you a visualization of the spread of Angiv. So down here in Manoa Valley is where Angiv was first established. It was actually brought in in Tahiti by Harold Lyon in an area in the back of Manoa Valley known as the Lion Arboretum today. And that was over or almost 100 years ago in 1927. So all these decades, it had time to naturalize, and it made its way up through the Ko'olau mountain range. And basically this whole entire mountain range is suitable habitat for Angiv. So they do like these areas of high, rich water retention where all these streams and gulches and cliff sides. And I pointed out the Kaluanui Natural Area Reserve, or the Kaluanui NAR, because this is where I'll be focusing my presentation today. And just to give you a visualization of how far this spread actually went up the mountain range. So some of the methods of control that we do are the ground control, aerial mapping, and precision aerial spray. On the left is our fence unit of the Kaluanui NAR. So every natural area reserve in the state of Hawaii is fenced off, and it's just to keep out predators like pigs, goats, or even deer. So the ground control, because again, Angiv, not a lot is known about this plant. We did do some herbicide trials, and we partnered with the Ko'olau Mountain Watershed Partnership, or KMWP, to run some test trials. And after the study was done, we found that 100% Polaris AC Complete does effectively control this plant. And we do this method using an incision point application, or an IPA. So ground control in Kaluanui NAR did start in 2017, and it is our newest NAR, so we didn't know really what to expect or how the terrain was. So we basically went in blind, but all we knew was that there was plants in the area. So how we do tree our plants, on the left side is where we first start. So what you want to do is cut off the fronds. And this plant is actually about the size of my hand. It's considered a small plant, maybe just a little bigger, but it's still small. So we base the sizing off the brain or the rhizome of the plant and not the fronds. So first we cut off the fronds. And then in the middle here, you see me doing the IPA. So it was a small plant. I just did 3 incisions. So 1 at the top of the brain and then 2 on the sides. And then on the left here, I have our syringe gun, and it holds about 5 milliliters of herbicide. And this plant only needed about 3 to 5 milliliters of herbicide. And you want to distribute that amount evenly through the different cuts. So I distributed about 5 milliliters evenly throughout the 3 cuts I did on this plant. And another visualization of how big these plants do get. So this is KMWP. They're doing some ground control. And this plant you see here, I don't know if you can notice the brain under the vegetation, but this is about 2 feet in length and width, and they do get really big. They get about up to 3 feet in length and width, and that's just the brain. The fronds can get up to about 12 feet in length. So some of the problems that we encountered doing our ground control was the steep terrain. Because we went in not knowing what we were getting ourselves into, we did find ourselves binoing a lot because of how cliffed off these areas were. There were a lot of natural barriers. So this NAR is considered our stream NAR. So we did run into a lot of waterfalls and areas that we just could not contour around. And sometimes these plants are just too big to control. They get huge to the point where we just don't have enough herbicide or enough staff to properly control every single plant in an area. So some things to help us mitigate this is our aerial mapping. So we hired a private company, Resource Mapping Hawaii, and they specialize in digital area mapping and remote sensing. So what they did was basically flew over a plane over the Ko'olaus. They gathered some of the digital mapping for us, and then they sent us all the files. And then Dylan Davis from KMWP, our partner program, he put it all together, put a map that we call the RMH Online and Analysis. And what he does is he'll overlay the RMH layer, and this basically helps us identify plants before going in to do ground or aerial work. So what we do is we'll set up a grid. So the white meaning it wasn't surveyed yet, and yellow meaning it was. You'll zoom into a plot, and then the closer that you zoom in, the RMH layer, the digital aerial map layer will kick in. And then from there, you can identify the plants. So you'll mark it, and then you'll fill out the attributes. So the management unit that you found it in, the date it was observed, the species, how much number or the number of plants identified, if it was detected or treated, mature, immature, and the action that you found it. So at the end of the day, we get this beautiful map, and most of these points were found through that RMH layer or that digital mapping layer. And the red just indicates that those are plants that are still detected, and the green meaning that they went in and treated. So some of the problems that we encountered doing this aerial imagery work was the plant identification. So there are some areas where it's definitely known that there's plants in the areas you see the huge, beautiful, lush fronds. And then we get into some areas that are very sparse or that are high in canopy, so the plant isn't always visible. And sometimes doing the analysis to see each plot and go through each plot does take a lot of staff time. So after doing that imagery and doing all the points, we can then move on to our precision aerial spray. And we use this to do to control high density areas or in areas that are too inaccessible or too remote to reach by ground. And because we do this at a more foliar spray, that the herbicide mix is actually a lot less. So the ground control was just at 100% imazapyr, whereas our mix for the spray ops is only at a 2% with a 1% surfactant. So here I have our spray rig, and it was actually made by Kyle Dorn, who is the owner of the company that we contract out to do some of our conservation work. So he got together with his mechanic and he built this little rig for us, and it costs about less than $1,000 to make, and it holds about 25 gallons of herbicide. So what he did was he found just like an old propane tank, he welded it, added the electrical wire, the electrical pump, and 4 nozzle sprayers at the bottom. So after connecting the spray rig to the bird, they'll go in and fly and do the aerial spray. On the right over here, the picture isn't what we do for the Angiv, but it is the same type of aerial spray, just with a different species. And it's just to give you an idea of what it looks like. So instead of this whole cliffside or pulley side being dyed in blue, it's actually all these little spots are specific plants that they go and hover over to spray. So at the end, how effective is it? It is very effective. So on the left are bunches of Angiv, healthy, mature plants. And then we went back in a year later to take a photo. And as you can see, they are all dead. You can see the old frond stalks laying by the dead brains. And then there is actually regeneration of native plants coming back in. So some of the problems that we encountered doing this aerial spray is the herbicide application rate, because we don't always use the same pilots. When we tell them hold the button between 2 to 3 seconds can vary between the pilot. And then we do run into mechanical issues. Maybe there's something wrong with the pump, or the electrical wire is has a little defect. And then the plant identification with the pilot and their knowledge of the area, because we can't physically be in the bird while the spray rig is attached. We do have to do a flyover before the aerial spray happens, and we do some plant identification with them, and we do hover over areas that we want to focus on for the day. And then on the right that you see here is plants that were mistreated, and on the top it was actually a native haupu'u fern, and then on the bottom you see there is a treated Angiv, and these were both treated during an aerial spray op. So at the end result, we do have positive notes that we did, in fact, treated over 1,000 plants within or around the Kaluanui Nar. And the good thing about these plants is it does take a while for them to grow and mature. They take about 3 to 4 years into maturity, which helps with our monitoring. So we do go back in with a drone to monitor or to fly back after an aerial spray has happened, just to see if there were any plants that were not treated or left that we can possibly do on ground. And then here on the right is a picture of our gigapan. So a gigapan is basically stitching a whole bunch of photos or series of photos together, and then it will give you this beautiful panorama, and you can zoom in at a high quality and kind of pinpoint these plants. And because it takes a few years for them to mature, I can go back on a year-to-year basis to see if there is any regeneration happening. So what's next for this project? We've started our Angiv specialized volunteer program, where we work with the community who do want to come out on a regular basis and kind of help us control and map Angiv in our high priority conservation areas on the island of Oahu. So on the left is just showing you our high priority areas for our conservation zones. And these are the areas that we want to have our volunteers help us focus in so that we can get this plant under control. Thank you everyone for tuning into my presentation. If you have any comments or concerns or questions, please feel free to contact me. Or if you have any data or GIS questions, please contact Dylan Davis from KMWP. Mahalo nui loa. Thank you so much everyone for tuning in. I hope you have a good day.